reciprocity of effects between Th1 and Th2 cells. Th1 cells produce interferon gamma that inhibits Th2 cells. Th2 cells, they produce interleukin-10 that inhibits Th1 cells. What is the immunologic advantage of having this reciprocal relationship? This is a good diagram to explain this relationship. Th1 cells are more adapted for cell-mediated immune defense against intracellular pathogens. Th2 cells, on the other hand, are more adapted for humoral immunity and targeting extracellular pathogens. Given that at any point of time, the top offending pathogen is either an intra or extracellular pathogen, this reciprocal inhibition would allow for a more vigorous production of the cell types that are specialized to destroy that particular offending pathogen that at that particular time has caused problems for the host. For instance, if the APC cells deliver information about extracellular bacteria to the lymph nodes, they will activate the Th2 cells. These cells will then activate B-cells to produce plasma cells and perform immunoglobulin switch for destroying the offending extracellular bacteria. At the same time, the interleukin-10 that is produced by these cells will shut down production of Th1 cells as they are not needed for defense against extracellular pathogens anymore. What immune cells are specialized in causing apoptosis of neoplastic cells? I'm going to be referring to this diagram. I'm showing a CD8 positive T cell to you. Cytotoxic T cells are also known as CD8 positive T cells. In addition to cancer cells, destroy cells that are infected with viruses and allografts, such as transplanted kidney, heart, and lungs. When exposed to infected nucleated cells, TC cells release their cytotoxic compounds, namely perforins, granzymes, and granulizines. Perforins make holes in the cell membrane through which granzymes enter the cytoplasm of the target cells and eventually cause apoptosis of the cells. I want you to note that granzymes are serine proteases that induce programmed cell death in the target cells that are cancerous or infected with viruses or intracellular bacteria. Another way in which cytotoxic T cells cause apoptosis of infected cells is by Fas ligand Fas killing. Fas ligand, or Fas-L, is a transmembrane protein. I'm showing the leg of it getting inside the CD8 T cell to show you that this is a transmembrane protein. Most nucleated cells express receptors for Fas ligand, which are designated as Fas in this diagram. When cytotoxic T cells bind to their targets, they produce more Fas ligand molecules on their surface. These Fas ligand molecules bind with the Fas on the target cells and cause their apoptosis. So there are two ways that the apoptosis happens. One is by granzymes and perforins, and the other is by Fas ligand Fas interaction. So let's talk a little bit more about Fas ligand and the concept of programmed death. Fas ligand, which is designated as Fas-L or CD95L, belongs to the tumor necrosis factor family. Upon binding to its receptor, which happens to be the FAS or FAST receptor or CD95, it induces apoptosis. FAST ligand FAST receptor interaction plays an important role in immune response to intracellular infections and control of progression of cancer. What receptor related to tumor necrosis factor binds to FAST ligand and prevents FAST ligand interaction and blocks the programmed death of the cells? And of course, as you can tell, if you block the programmed death, the cells can become potentially immortal, or they may potentially constantly divide, like the cancer cells. The answer to the question is DCR3, or decoy receptor 3, and I'm showing this as a circle attached to FAST ligand on CD8 T cell. This protein is a soluble receptor that has no signal transduction capabilities. Therefore, we call it a decoy, and prevents FAST receptor FAST ligand interactions by competitively binding and inactivating the membrane-bound FAST ligands, as you see in this diagram. Overexpression of DCR3 is postulated to be involved in induction of angiogenesis and certain gastrointestinal tract tumors. So, of course, angiogenesis involves continual growth of the vessels, and tumors involve continual division of the cancer cells. So these are some of the functions of decoy R3, or decoy receptor 3 protein. Macrophages, likewise, dendritic cells are antigen-presenting cells. However, they are by far the most potent phagocytes of the innate system. What roles do interleukin 2, 4, and 12 play in the T cell, B cell, and macrophage interactions? I'm going to be referring to this diagram for you. Macrophages, likewise dendritic cells, express major histocompatibility molecules and pathogenic epitopes. Encountering of the pathogens activates macrophages, and they produce interleukin 12 and other cytokines. And then they migrate, likewise, dendritic cells to the nearby lymph nodes to encounter their cognate naive CD4 positive T cells and B cells. Interleukin 12, which is produced by the antigen-presenting cells during infection, primarily regulates innate responses and determines the type of adaptive immune responses. Interleukin 12 induces interferon gamma production and triggers CD4 positive T cells to differentiate into T helper 1 cells, and they in turn make interleukin 2 and interferon gamma, and they further activate macrophages. In contrast to T helper 1 cells that mainly secrete interleukin 2, interferon gamma, and interleukin 12, the Th2 cells secrete interleukin 4, 5, and 10 that promote humoral immunity and B-cell activation. It is strongly believed that the balance between interleukin 12 that favors Th1 or cell-mediated responses and interleukin 4 that favors T helper cell 2 or humoral responses determines the early preference expressed in the immune response. A patient is diagnosed with ascariasis. What is the most efficient adaptive immune response to the patient's disease, and how is it coordinated. Of course, I'm going to be referring to this diagram in explaining the concept for you. The immune response against most helminths includes Th2 helper cell response that is associated with increased levels of interleukin-4, plasma cells that produce IgE, and eosinophils, mast cells, and basophils. So let's summarize. The major characters involved in helminthic diseases are IgE producing plasma cells, eosinophils, mast cells, and basophils. By contrast, interferon gamma and Th1 type responses are classically induced by bacterial and viral infections, and they are associated with Th1 cytotoxic CD8 positive T cells, neutrophils, and macrophages. So let's summarize again. Th1 cells, neutrophils, macrophages, they go with the stimulation by interferon gamma. In this particular diagram, in contrast to the diagrams that I've shown before to you, I'm showing a B cell as our antigen presenting cell. Remember, B cells can also act as antigen presenting cells, and this B cell is reacting to the antigen of the ascaris. And I'm showing that in event number 1. The B cell, of course, exhibits an epitope of the antigen of the ascaris on its surface MHC2 molecules. Meanwhile, the co-stimulation by B7-CD28 interaction accounts for the co-stimulatory response. This is event number 3. This interaction activates the T helper 2 cells, and they produce several cytokines, including interleukin-4. Interleukin-4 now acts on the naive B cells. I want you to note that for illustration purposes, I've shown the original B cell in this diagram, but you can imagine that they can act on other B cells. And other B cells, they get activated and produce pathogen, a specific response that includes isotype switching from membrane-bound IgM to IgE. And this is event number 5. And conversion of the B cells to plasma cells. Plasma cells that they have the ability to produce high titers of IgE antibodies against the antigens of the ascaris parasite. These are event 6 and 7 in this diagram. Let's look at the cytokines that they have the ability to induce class switching. And let's look at the most important associations. T cell types that we're going to be looking at are Th2, Th1, and Tregs. Cytokines produced by Th2 are interleukin-4 and 5. Interleukin-4 increases IgG1 and IgE, and it reduces the IgG3 isotype. Interleukin-5 does one thing, and does it greatly. It increases the IgA production. Th1, the cytokine that it produces, is interferon gamma. Interferon gamma decreases IgG1 and IgE, and at the same time it increases the IgG3 production. So in a sense, interferon gamma acts exactly opposite to interleukin-4, which is produced by Th2 cells. Treg cells produce tumor growth factor beta and interleukin-10. Tumor growth factor beta decreases IgG3 isotype and increases IgA. Interleukin-10 increases IgG1 and IgG3 isotypes. What is the mechanism and what steps are involved in antibody isotype switching? And as you can tell, I'm going to be referring to this diagram for you. And we're going to follow the diagram from top to bottom. I'm showing the 5' end of the DNA on the left side. I'm showing the VDJ segments followed by the mu chain, D chains or delta chains, gamma 3 or G3, gamma 1 or G1, A1 or alpha 1, G2 or gamma 2, and G4 or gamma 4, followed by epsilon or IgE, and finally IgA segments. Have that in mind. Let's go through this together. B cells must be activated by a specific T cell cytokines that trigger formation of proper antibody isotypes against the pathogen. For instance, interleukin 4 and 5 promote IgE and IgA formation respectively. Class switch recombination, CSR, changes the class of antibody of an activated B cell. During class switch recombination, portions of the antibody heavy chain locus are removed from the chromosome, and the gene segments surrounding the deleted portion are rejoined to retain a functional antibody gene that produces antibody of a different isotype. The diagram shows how to switch the original membrane-bound IgM isotype, which is the original isotype, and I'm showing it on top, to Ig3 isotype and discard the intervening mu and delta segments. In the activated B cells, there is an enzyme known as 8 enzyme. Stands for activation induced deaminase, 8 enzyme. This enzyme breaks the constant region of the heavy chain. Double-stranded breaks are generated in DNA at certain points that are known as switch or letter S points. Switch points designated with letter S. These regions are immediately upstream from the gene segment that encode antibody isotypes. The DNA is then nicked and broken at two selected S regions in such a way that part of the switch interons are upstream from the target gene segments. The remaining part of the switch intron on both ends will provide sticky ends for splicing the two segments and filling the gap. Meanwhile, the unwanted segments in the diagram, the mu and delta DNA segments, are deleted from the chromosome, and DNA repair enzymes now step in and construct a new constant region for us. Let's talk a little bit more specifically about isotype switching. Mature naive B cells start out with IgM and IgD isotypes on their surfaces. These are membrane-bound antibodies. However, after encountering their cognate antigens, they will lose their IgD and turn into IgM secretors. Many of these IgM-producing B cells later switch their IgM to other forms of immunoglobulins that are more suited to handle the very same epitope or the very same pathogen in various tissues. If B cells receive a specific signal from T helper cells or their environment, they can undergo antibody class switching to produce other isotypes that are more effective against the encountered pathogens. During class switching, the B cells can switch from production of an upstream isotype on their heavy chain to a downstream isotype. For instance, IgM can switch to IgG or IgA, and IgG can switch to IgA and not vice versa. no downstream isotype may change to an upstream isotype. For instance, IgG cannot switch to IgM. In other words, switching is top to bottom unidirectional. It is 5' to 3' directional. Switching only changes the constant heavy chain, and the light chain and the variable chain of the antibody remains unchanged. In this process, the B cells maintain their antigen epitope specificity. That is, the idiotype is going to be kept constant, but it gains the advantage of binding the same epitope by different isotypic antibodies. In other words, switching will only modify the effector functions that each class of antibody has to perform. Isotype switching allows for progenies of the same activated B cells to produce antibodies of different isotypes against the very same epitope or antigen or peptide. Just note that effector functions of an antibody may simply be defined as how effectively a form of an antibody can eliminate an antigen. For instance, IgA is great for attacking the antigens on mucosal surfaces. IgG is great for attacking the very same antigen in the tissues and in the blood. What are the main immunologic advantages of isotype switching? We have already answered part of this question, but let's look at it more comprehensively now. Switching allows for progenies of B cells which will be able to produce isotypes that have discrete sets of biological activities. For instance, a mother who has acquired immunity to rubella may pass along only the IgG idiotypes against rubella through placenta to her newborn, because this is the only isotype that can pass through the placenta. The IgA idiotypes are more suitable against mucosal diseases caused by a particular antigen, whereas IgG idiotypes are more suitable for tissue infections. What is the descriptive term for the process by which T-cell activated B cells produce antibodies with increased affinity for antigen during the course of an immune response and after repeated exposure to the same antigen? Affinity maturation. In this process, subsequent generations of the antibodies will progressively exhibit greater affinities for the antigen. Affinity maturation primarily occurs in the germinal centers of the secondary lymphoid organs. It involves two interrelated processes of somatic hypermutation and clonal selection. What is the term that best describes the occurrence of point mutations in the exons of the variable regions of an immunoglobulin molecule during the course of immune response that tends to increase antibody diversity and antigen binding affinity? Somatic hypermutation. Do not mistake somatic hypermutation with somatic recombination. Somatic recombination refers to VDJ recombination. Following activation with antigen, B cells proliferate rapidly, and the genes encoding for the variable domains of the heavy and light chains may undergo a high rate of point mutations. These mutations often change only one nucleotide in each variable gene for each cell division and lead to daughter B cells with slightly modified variable domains and slightly different affinity for the antigen. Somatic hypermutation increases the overall diversity of the antibody pool and impacts the antibody's antigen binding affinity. Note that some point mutations, as you can expect, will randomly result in production of antibodies that have a lower affinity for their antigens. However, I want you to know that these B cells will not receive positive survival signals and will be eliminated by apoptosis. In contrast, point mutations that lead to a higher affinity for the antigen will receive positive survival signals. Let's look at the three major mechanisms involved in antibody diversity. One is random recombination of VJ segments of light chain and VDJ segments of the heavy chains. Remember, as we said earlier, light chains are lighter, they miss one segment. They miss the segment D. The second phenomenon is affinity maturation and somatic hypermutation, and the third is isotype switch recombination. Now let's turn our attention to B lymphocytic development in bone marrow. I'm showing also the immunoglobulin types that are produced during the B cell development. On the left column, I'm showing the cell types, beginning with hematopoietic stem cells. They will not express any immunoglobulins. Just to know, hematopoietic stem cells, lymphoid cells, and pro-B cells, none of them express immunoglobulins. Pro-B cells, however, express partial heavy chain rearrangements. Pre-B cells express mu heavy chain, which is the IgM essentially chain, and also surrogate light chains. The pre-B cells are produced after pro-B cells undergo complete heavy chain rearrangement. From pre-B cells, we progress to immature B cells. Immature B cells express membrane-bound IgM, and these cells are produced after pre-B cells undergo complete light chain rearrangements. Finally, from immature B cells, we progress to mature B cells that they have membrane-bound IgM and IgD. So immature has IgM only, mature B cells, they have both IgM and IgD that are membrane-bound. Again, remember the progress from pro to pre to immature to mature. This is something that historically has confused many medical students. Let's look at the B cell development in peripheral lymphoid tissues. The types of the cells that I'm going to be considering are mature B cells, activated B cells, IgM secreting plasma cells, and we're going to be looking at the concept of class switching and memory B cells of all isotypes for the same idiotype. Now, mature B cells express membrane-bound IgM and IgD. Activated B cells are produced after antigenic stimulation of the mature B cells. IgM secreting plasma cells are produced after differentiation of activated B cells. Class switching starts with IgM producing cells. Memory B cells, plasma cells produce all different types of isotypes for the same epitope or idiotype. That is, they produce IgG, IgA, IgE against the very same pathogenic antigen or epitope. Immature B cells express membrane-bound IgM, and when they mature, they express membrane-bound IgM and IgD. At what time in their development do they lose their membrane IgD? After their antigenic stimulation, that is, after the first encounter with their cognate antigen. Where does this happen? This happens in the secondary lymphoid tissue. Mnemonic: B begins in a bursa or in a bone marrow. B lymphocytes perform many immunological functions. What is their most straightforward definition? These are cells that express clonally diverse cell surface immunoglobulin receptors for recognizing specific antigenic epitopes, and each cell produces clones of one and only one antigenic determinant. This diagram explains it. I'm showing two different B cells. Each one of them produces one specific type of antibody against a specific epitope. Where does B cell development begin and where does it finally end? It begins in the primary lymphoid tissues with final maturation in the secondary lymphoid tissues. So what are primary lymphoid tissues? These are sequentially fetal liver, fetal bone marrow, and adult bone marrow. What is by far the most important functional developmental feature of the B cells? Rearrangement in the immunoglobulin site. These rearrangements happen in the fetal liver and adult bone marrow and involve random combinations and rearrangements of V, D, J segments of the heavy chain and V and J gene segments of the light chains. Final outcome is a diverse repertoire of functional VDJ rearrangements for the heavy and VJ rearrangements for the light chains that encode the B cell receptors. What two enzymes play an exceptionally important role in generation of diversity in the B cell receptors? The recombination activating enzymes, RAGs, that are encoded by RAG gene. There are two major RAG genes that we have identified, RAG1 and RAG2. These genes are only expressed in lymphocytes during their developmental stages. RAG proteins play an important role in the rearrangement and recombination of the genes of immunoglobulins and T-cell receptor molecules. The diversity of immunoglobulins is the outcome of shuffling, cutting, and recombining a few hundred genes, the VDJ genes, to create millions of permutations in a process called VDJ recombination. RAG1 and 2 enzymes separate, shuffle, and rejoin the VDJ genes during their maturation. Let's talk a little about membrane-bound versus secreted immunoglobulins. A naive B cell only expresses monomeric and membrane-bound IgM. Lock in this information. At times, it may also produce membrane-bound IgD in addition to the IgM. After stimulation by its cognate antigen, the B cells begin to produce and secrete pentameric, which is secretory version of the IgM molecules, into their surroundings. The mu heavy chain of the membrane-bound IgM has an amino acid sequence at its carboxyl terminal that anchors the immunoglobulin into the B-cell membrane. The carboxyl terminal of the secreted form lacks the anchoring region. This diagram shows the carboxyl end and shows how the membrane-bound IgM, which is a monomer, is attached to the B cells or to the B cell membrane. The membrane-bound IgM is also unable to combine with J chain to make a pentameric form. As the B cell develops into an antibody-secreting plasma cell, it may transiently produce both membrane and secretory form of the IgM, and finally only the secretory form of IgM. A memory B cell which synthesizes only membrane-bound IgG will change to producing only the secretory form of IgG as it develops into a plasma cell after secondary antigenic stimulation. Note that mature immunoglobulin molecules are secreted from the plasma cells by exocytosis of post-Golgi vesicles. Exocrine secretion of IgA into GI tract, respiratory surfaces, tears, and saliva is a unique process that requires a special secretory component or S component that I'm showing it in purple in this diagram. This component is added to IgA by secretory epithelial cells. So secretory component is produced by epithelial cells to this immunoglobulin. And of course, I'm showing also a joining, a J chain that connects the two monomers of the IgA for you. So IgA that is on the B cells is monomer. However, the IgA secreted into tear or secreted into the respiratory surfaces is a dimer. So what unique feature of the secreted form of IgE makes it distinguishable from other immunoglobulins? First and foremost, concentration of IgE in the serum is the lowest of all immunoglobulin subtypes, and it also has the shortest half-life of all, about 6 to 32 hours. Secondly, the tail region of IgE molecule binds with extremely high affinity to the surface Fc receptor of their effector cells, namely mast cells in tissues and basophils in the blood. This happens immediately after IgEs are released from the plasma cells. Finally, expression of IgE is strongly regulated in the absence of infections. What unique feature of IgE makes its effector cells similar to B cells? IgE molecules that bind to the surface of mast cells function as passively acquired receptors for antigen. Antigen binding triggers these effector cells to secrete a variety of cytokines and biologically active amines, especially histamine. Note that IgE is abundant in epithelial and mucosal tissues and attaches to receptors of the tissue effector cells such as eosinophils and mast cells. The bound form of IgE has a very long half-life, up to several months. This fact confirms that IgE must play an important role in local immune defense mechanisms. Also, I want you to note that IgE is one of the most evolutionary conserved immunoglobulins. It is claimed to have evolved over 250 million years ago, and its presence in the mucosal cells also supports the fact that innate immune system is by far the most primal of the two immune systems. What are the essential components of an allergic inflammation? IgE, mast cells, basophils, and eosinophils. What is the key step in initiation and propagation of immediate hypersensitivity reactions? Antigen-specific IgE production with subsequent fixation to FcRI receptors on mast cells and basophils. I'll be talking about Fc epsilon RI receptors in a second for you. Cross-linking of FcRI via IgE antigen complexes leads to degranulation of mast cells or basophils and release of inflammatory mediators. Note that mast cells, basophils, and eosinophils are central effector cells in allergic inflammation as well as in innate and adaptive immunity. Finally, note that the high affinity IgE receptors, abbreviated as Fc epsilon Ri, is the receptor for the Fc region of the IgE. I want you to know we also have another type of receptor known as low affinity Fc epsilon receptor that is also known as CD23 or Fc epsilon R2 that is expressed on resting B lymphocytes, monocytes, macrophages, eosinophils, and platelets, but rarely on the T cells. What interleukins play important roles in immunoglobulin class switching of the B cells and IgE production? Isotype switching to IgE requires two signals. The main signal, or signal 1, is provided by interleukin 4 and or interleukin 13. These signals act through the interleukin 4 and interleukin 13 receptors and activate transcription at the IgE isotype specific switch region, abbreviated as S epsilon switch region. Note that signal 2 is provided by ligation of CD40 on B cells by CD40 ligands on the T cells, which activates DNA switch recombination. What is STAT6 and what role does it play in immunoglobulin class switching? So as you can tell, this protein, STAT4, has a lot to do with class switching. I'm going to be referring to this diagram for you. STAT6, also known as signal transducer and activator of transcription 6, is a protein involved in translocation of signals to the nucleus of adaptive cells, and it is a transcription activator. This protein plays a central role in mediating interleukin 4 signals and thus in B cells IgE class switching. Note that the process of class switching of IgE is initiated when allergen is taken up by antigen presenting cells such as allergen-specific B cells. Epitopes of the allergen are then presented in the context of MHC class 2 to Th2 cells that they recognize the allergen MHC2 complex. Activation of the allergen-specific Th2 cells leads to expression of interleukin 4 and 13 and a stimulation of the B cells and induction of class switching to IgE. Note that originally class switching begins with the T cell activation of the B cells. However, basophils are also able to express high levels of interleukin 4 and interleukin 13, and they play roles in later IgE production. Also note that mast cells are able to fixate a high number of IgE molecules to their high affinity FcRI receptors, but unlike the basophils, they do not produce interleukin 4 or interleukin 13. So they act as a reservoir for IgEs that are formed by the plasma cells.